Fish have an immune system that protects them from disease and environmental stress. Unlike mammals, fish rely mainly on spleen, kidney, and liver to produce immune cells, fight pathogens, and regulate immune responses. In this practical, these organs were identified through dissection, tissue smear preparation, gem staining, and microscopic observation to understand their structure, immune cell, and important roles in maintaining fish health. The objectives are to identify and examine the spleen, kidney, and liver through anatomical observation and dissection, to observe the tissue structure and identify the different immune cells present in the spleen, kidney, and liver using gemstone tissue smear and microscopic examination, and lastly, to compare the morphology and cellular characteristics of spleen, kidney, and liver and understand their respective roles in fish immune system and overall health.
Next is materials. We use fresh fish, microscopes, microscope slides and cover slips, sterile saline solution, dissection tools such as scissors, forceps, scalpel, and tray, and the last one is gem stain.
Moving on to procedures. Step 1. Preparation. First, wear a lab coat and glove to ensure safety. Next, all the materials and dissecting tools were prepared before starting the dissection. Step 2. Fish dissection. The fish was carefully dissected to expose the internal organs. The body cavity was opened gently to avoid damaging the lymphoid organs. Step 3. Identification of lymphoid organ. The organs spleen, kidney, and liver were identified. The kidney is located along the dorsal side of the body cavity. It is the main hematopoietic organ responsible for producing blood cells and immune cells. Next, the spleen is a small dark red organ located near the stomach. It filters blood, removes damaged blood cells, and stores immune cells. Lastly, the liver is located in the abdominal cavity near the digestive organ. It helps support immunity by filtering blood, removing pathogens through immune cells, and producing protein involved in immune defense. Step 4. Smear preparation. A small amount of spleen, kidney, and liver tissue is placed on the clean glass slide. The tissue is gently smeared to create a thin layer, then allowed to air dry before staining. This process helps to observe and identify immune cells under microscope. Step 5. Fixation. The smear was air-dried and fixed with methanol for about 3 minutes. Fixation preserves the cells and prevents distortion during staining. Step 6. Staining. The smear was stained using red gemstone according to the laboratory procedure. After staining, the slide was gently rinsed with distilled water and allowed to air dry. Step 7. Microscopic observation. The stained smear was observed under a light microscope. Observations start with low magnification to locate the cells, followed by higher magnification to examine the cell's morphology in detail.
Microscopic observation of the gemstone smears reveal differences in the cellular composition of the kidney, liver, and spleen. The kidney showed a high density of hematopoietic and immune cells, reflecting its role as the primary lymphoid organ in fish. The liver contained fewer immune cells and was mainly composed of hepatocyte-like cells, consistent with its function as an immune-associated organ. The spleen exhibited abundant nucleated erythrocytes with scattered immune cells supporting its role in blood filtration and immune surveillance. Overall, the differences observed among these organs demonstrate their distinct but complementary functions in maintaining the fish immune system and protecting against pathogens.
In conclusion, this practical successfully achieved its objectives by identifying the major lymphoid organs in fish and demonstrating the correct techniques for preparing, fixing, staining, and observing lymphoid organ smears. The kidney, spleen, and liver were identified as important immune organs, each playing a vital role in the fish's defense against pathogen. Through smear preparation and right gymnastic staining, lymphoid cells were clearly observed under a light microscope, allowing the morphology to be examined and interpreted. This practical enhanced our understanding of the structure and function of fish lymphoid organs while developing essential laboratory skills in dissection, cytological smear preparation, staining, and microscopic observation. Overall, the knowledge and techniques gained are valuable for fish health assessment, disease diagnosis, and aquaculture research.